Good afternoon. I'm Cammie Abernathy, Dean of the Herbert Wertheim College of Engineering. Thank you for joining us today as we have an exciting presentation planned for you. We're thrilled to have Cherie Stabler to discuss the challenges and recent advancements in engineering cellular implants for treating type 1 diabetes. Dr. Stabler is a professor and Integra Life Sciences term professor in the J. Creighton Pruitt Family Department of Biomedical Engineering at the University of Florida. She's also an affiliate member of the Diabetes Institute at UF's College of Medicine and an adjunct professor at the Diabetes Research Institute and the Department of Biomedical Engineering at the University of Miami. She's an American Institute for Medical and Biological Engineering Fellow and the recipient of the 2008 NIH NIDDK Type 1 Diabetes Pathfinder New Innovator Award. She received her PhD in Biomedical Engineering from the Georgia Institute of Technology and Emory University. She completed her postdoctoral research supported by a JDRF postdoctoral fellowship under the guidance of Dr. Elliot Chaikoff. Her research centers on the engineering of cell-based tissues for the treatment of type 1 diabetes, specifically the development of novel biomaterials for cellular encapsulation, three-dimensional scaffolds, and in situ oxygen and drug release. Through the fabrication of novel biomaterials capable of actively interfacing with the host, she seeks to modulate the graft environment to favor the survival and optimal function of the implanted cells. Her laboratory works across the spectrum of research from polymer development to benchtop studies and preclinical models to clinical translation. She has published in a broad range of journals, from the Proceedings of the National Academy of Science to Biomaterials to ACS Applied Materials and Interfaces to Transplantation. With that, I will hand it off to Dr. Stabler to begin the presentation and discussion. Dr. Stabler.
All right. So I'm thankful for the opportunity to talk today about some of the work that we're doing in engineering treatments for type 1 diabetes. So whenever an opportunity to speak to a broader audience about the work that we do, I like to take the opportunity to talk about type 1 diabetes because there's a lot of misperceptions about this disease because diabetes encompasses a broad spectrum of different types of conditions and pathology. So type 1 diabetes is approximately 5 to 10% of diagnosed diabetics in the United States. And so the vast majority of diabetics in the US are type 2, which has a different pathology. Now, type 1 diabetes, which used to be called juvenile diabetes, and it stopped being called that because really it can occur at any age. It most often develops in children and young adults, but we're seeing a larger cohort or a growing cohort of people that are being diagnosed at later ages, sometimes up to their 40s. It is an autoimmune condition, so it is not something that's precipitated by poor diet or poor care. It is really your immune system, for some reason, recognizes specific cells in your body as being foreign and attacks and kills those cells. So just to give a brief overview of what these cells are. So in the pancreas, I hope you can see my mouse. In the pancreas are these little islands of clusters of cells called pancreatic islets. And these pancreatic islets comprise several different types of cells, but they secrete essential hormones that are important for physiological regulation. In particular are these beta cells, and these beta cells are responsible for secreting insulin. And insulin is kind of a key to get glucose into your cells. So without insulin, it's actually a lethal condition. So prior to the discovery of insulin, anyone that was diagnosed with type 1 diabetes actually passed away. It was a lethal condition because they didn't know what was causing it. Once they discovered insulin, and insulin was the cause of this, they found that in type 1 diabetes, this autoimmune condition kills these beta cells. So these beta cells are destroyed. The body no longer produces its own insulin. And it requires the need for exogenous insulin, exogenous being, outside of the body. So they have to receive these injections or pumps. And so this is a treatable condition. It's one that they can, move from a lethal to a treatable condition. And this is really a medical marvel, a substantial discovery to really shift the lives of people suffering from type 1 diabetes. But it's not not without cost. People that have type 1 diabetes really struggle with the mental, economic, and physiological burden of this disease. So while they can take exogenous insulin to manage it, and there's significant improvements in terms of the delivery of insulin. So there's now insulin pumps as well as glucose sensors, these continuous glucose sensors that you can place on under the skin and that can continuously sense the blood sugar levels or, levels in the body. This does not alleviate that mental, physiological, and economic burden. You envision somebody that wants to have a meal, sit down and have something to eat. A person with type 1 diabetes has to estimate, well, how much insulin would that require? How much insulin should I give myself now? And they constantly have to make these decisions about whether their blood sugar should go slightly high or slightly low. If they go too low, this becomes a lethal condition. This increases their chances of death. But if they go too high, it substantially increases the challenges and complications of high blood sugar, which is chronic wounds, kidney challenges, eye challenges, cardiovascular disease, and other things. And so this is a really challenging life of people with type 1 diabetes. And for a selected cohort of type 1 diabetics, even with appropriate management, really trying their best to manage their insulin levels, they are struggling to maintain stable normal glycemia, so stable glycemic levels. So this is an example of a continuous glucose monitor for a patient that is under endocrinologist care, and you can see over the course of several days, their blood sugars are oscillating. And this is what causes these challenges of side effects and also challenges of, if they go too low, really complications, severe complications. So for these patients, for these people struggling, they are eligible for what we call clinical islet transplantation. And what clinical islet transplantation does is it replaces those cells that are lost. It basically isolates islets from a cadaveric donor, pancreas. And then these clusters of cells are the portal vein of the liver, and they basically fly through the bloodstream until they stick and they just can't move any further in the bloodstream. And they then sit there and secrete insulin in response to glucose. And this seems kind of a brutal method, but what's amazing is that it actually works very, very well in terms of secreting insulin and managing blood sugar. So this is the same patient pre and post transplant. And you can see from this very stable blood sugars. And if you showed this to an endocrinologist, they would not know that this person has type 1 diabetes. And so this is really, this potential cell therapy is what drives me in my laboratory every single day, is that we could find a long-term treatment option for people suffering with type 1 diabetes, in which they wouldn't have to make those mental decisions anymore. These cells would inherently know how to regulate their blood sugar and secrete the appropriate amount of insulin. So why isn't everyone receiving this? Many of you probably can see some of these limitations already. One is it's reliant on a cadaveric donor. And so anytime you're relying on organ donations for the procurement of your cell source, there's a limited source for that. The other is that it's a non-durable therapy. And so if you're an optimist, maybe you look over here and say, wow, you can take the most severely diabetic patients that are really struggling with blood sugars and achieve insulin independence. But maybe if you're a pessimist, you might look over here and say, well, it doesn't last very long. And this is true. There is a precipitous decline in insulin independence. And that's despite them still being C-peptide positive. C-peptide is actually a byproduct of insulin synthesis. So what this tells us is that the islets that were transplanted are still secreting insulin. It's just not enough to manage, to completely manage the blood sugar levels. The other challenge of this is that the patients have to be on immunosuppressive drugs. And immunosuppressive drugs, this is like an organ transplant, so it suppresses their immune system. And so you're certainly not going to take a 13-year-old who is remotely managing that pretty well, their diabetes really well, and give them a cell therapy that puts them on these immunosuppressive drugs for the rest of their lives. And so what we've learned from these clinical trials and what we've learned from patients undergoing these things is many, many things of why we get loss of these islets. And I won't go into all of these things because that would be quite overwhelming, but to break down kind of the critical issues of why this is not a durable therapy and why it's not a widespread therapy is, one, the transplant site is very poor. Just injecting them in the bloodstream of the liver causes a lot of problems. The other is immunosuppression. One, it is not one that the risk of immunosuppression is too high for most people. And also it's not completely effective. So over time, the graft does get rejected. And then finally, the cell source. And so to address this is really a multidisciplinary challenge. We need to move when we address these very broad medical issues. We need to approach it from a very multidisciplinary approach. If you think about it, this is a cell biology, a clinical issue. This is a immunology issue, and a lot of things to try and improve this. So why is an engineer talking about type 1 diabetes? So why am I here? Why is this engineer in the room? And what we can, engineers can provide in terms of addressing these clinical issues is really the engineering design process. So trying to ask questions, study what the problem is, try and imagine prototypes that would improve it. So just the basic engineering process, but also designing 3D implants. So similar to designing a building and building a building and scale up and manufacturing of those, this is what engineers can bring to that. So if the critical needs of improving cell therapy for type 1 diabetes is designing better implant sites and designing better cell implants, we can do that. We can really bring our engineering prowess to that and think about ways that we can do that, as well as some of these immunosuppression issues. And I hope to kind of highlight why an engineer is in this room and what we can kind of provide to address this challenge. So as I talked about, really approach the problem, do some research, understand the environment to develop prototypes. And so that's what we did, is first start with, why are islets in the pancreas? We're so beautifully designed for a reason. These islets are in very specific niches. They're in a pancreas for a reason. And what are the critical features so that we can think about how to replicate those? So what are the key design criteria that we want to think about? So one is islets are heavily distributed through the organ. So they only take up 1% of the organ, but they're really spread out through it. And also they're heavily oxygenated. So they have a lot of blood flow. They get 1.5 to 2-fold higher oxygen than the rest of the cells in the pancreas. And they also have a very, very high metabolic activity. Their metabolic activity is about 10-fold higher than that of your heart cells. You can envision your heart cells do a lot of work. Well, these islets need 10-fold more nutrients than that. They're also richly vascularized. There's all these blood vessels there. And there's other things that are very distinct about that. And so when we thought of trying to kind of engineer a better site, engineer a better environment for that, we looked at trying to create a way to distribute the islets in 3-dimensional space. So similar to like the scaffolding of a building that you see that coming up to create that 3-dimensional space, we created biomaterial scaffolds that would allow islets to be housed in a 3-dimensional space. And so this is our porous scaffold that we fabricated, and you can kind of sprinkle the islets in there and they'll distribute in a 3-dimensional space by kind of filling into these pores. It's a very mechanically compliant implant. So we tried to design the material one that allowed for compliance with tissue, so there wouldn't be this mechanical mismatch. And then the other thing we really strive for is where to put it. One of the things that is very exciting about islets is you can, in theory, put them anywhere, right? If you have to do a kidney transplant, you would put a kidney where the kidney is and hook up the plumbing, but you don't necessarily have to do that for an islet. But also you shouldn't just throw them anywhere. You have to think about that islets are in a pancreatic niche, richly vascularized. We can't just toss them under the skin and expect them to survive really well. So we actually spent an extensive amount of time identifying a site that we could transplant these scaffolds containing the islets. And what we identified was the omentum. And the omentum is this really cool tissue. It's kind of like an apron that hangs over your abdomen. And it's richly vascularized and has tremendous regenerative properties. And back in prior to about the 1960s, if you had severe trauma to your hand, what they would do, surgeons would do, is they would open up your abdomen, take out this tissue, wrap it around your hand, and then kind of suture your hand back into your abdomen, so intramuscularly. And you kind of walk around like Napoleon for like 6 weeks or so. And then they would remove it and they would see that it would help heal that hand. Now, obviously we have better methods now, but the omentum is really mobilized a lot by surgeons for breast reconstruction, for spinal, any sort of surgeries, bladder surgeries and things like that. They tend to mobilize the omentum and lay it over that site because they find it improves the efficacy of the surgery. And this is what omentum looks like this is a larger animal model. You can see the omentum here, it's richly vascularized, and these are little scaffolds put in there. We kind of wrap them up like a present. And this is explanted 445 days later. You can see it becomes richly vascularized. So we try and create this very open framework to let the host environment really remodel that space and vascularize that space. And we've done a lot of transplants and a lot of studies in preclinical models that show that this is very effective in treating diabetes in those animal models. The other thing that we're trying to do is further improve that site. So not simply just create this scaffolding, but start adding more things that would create a beneficial home. So if you think about creating a home for these islets, we want the framework of the house, but we also want to have plumbing and have nutrients be able to get there and things like that. And what we found is that when we take our scaffold and plant them in the omentum, it takes time for the vascularization to develop. And during this time frame, which can take about 14 days, the islets are suffering. They're not richly vascularized and they have very low oxygen levels. And so what we tried to do is actually create kind of like scuba tank for these scaffolds. So if there was a way that we could maybe create a material that would support and deliver oxygen during this very precarious time frame, this could be desirable. And so that's what we did, is we actually engineered and designed this material. We call it OxySite. And basically we have an oxygen generating material that when it is in contact with water or an aqueous environment, which is your body, inside your body, it will generate oxygen. And it can generate oxygen in a very durable manner. And we can create different prototypes and different geometries to really tailor and customize the oxygen release. And I'll show you some data just so you know I'm not making this all up. An example of using this OxySite disc in the center of an implant. So what happens typically is when you implant cells, the cells on the periphery of the device have the highest level of nutrients because they're closest to the blood vessels. And so as you can see here, this is a staining of cells distributed through this implant. Green cells are alive and red cells are dead. And you see on the outer edge, we have a lot of viable cells, a lot of green cells, pretty happy cells. But as we migrate deeper into the implant, you start seeing more and more dead cells. And that's because it takes a while for those nutrients to move through that, to diffuse through that implant, and that causes nutrient deprivation. So what we did is we put the OxySite disc in the center, that's our oxygen generating material, to serve as a supplemental oxygen delivery method. And what you can see here is that this then shifts that detrimental profile to be one in which all the cells are very viable. And this leads to improvement in efficacy of implants in preclinical models. So this is the OxySite treated ones. We get more of our animals become normal glycemic or are not diabetic anymore. Just to show you that there's a lot of data here, a lot of data I could show you, but I'm not going to show all of it. But just so you know that we've done a lot of studies on this. The other thing that we're really interested in is the immunosuppression side, is how can we customize or develop things that would allow us to mitigate the risk associated with giving immunosuppression to the entire patient. So this is the common way for organ transplants to be done, is you give them a repertoire of drugs that suppresses their entire immune system. So while the implant may be the heart, it's going to immunosuppress everything because the heart is connected, with blood and everything through that. But what we sought to do is maybe we can localize this approach to be one in which it's only at the implant site. So if we can create methods that will localize immunomodulatory or immune agents to that site, then the patient has restored, their immune system is fully functional. It's just kind of this protective bubble. So we can use much smaller drug loads. We can reduce side effects. We can decrease susceptibility for infections. And we can also use drugs that may not be able to be used systemically. And how we're doing that in our laboratory is through three primary ways. One is we're actually using the scaffold itself to deliver drugs. So we're embedding drugs within the scaffold and it's releasing that. We're also using a method called encapsulation. And finally, we're trying to use inspiration from natural mechanisms of immune responses to create materials that would dampen immune activation. So I'm going to briefly talk about these last two and give you a sense of how we're kind of developing those. So the concept of encapsulation is basically whenever you transplant something, whenever you transplant a kidney or a heart or an islet, the immune system recognizes that foreign implant through cell recognition. So an immune cell will come in and recognize that all these flags are different. This is not their cells, and then they'll reject it. So the concept of encapsulation is basically, let's just put a barrier, kind of like a moat around a castle, or creating a security system around that house. Or the analogy I like to use a lot is creating kind of a shark cage, a cage around the diver where all the sharks are around them, but the sharks can't get the diver. Now the cage must be porous enough to allow nutrients to come in. So we need to be able to have, nutritional thing, not necessarily hamburgers, but glucose and oxygen to be able to come in and feed the cells. And we also need insulin to come out. Otherwise, then why would we transplant this, right? But we don't want it to be too porous. That's detrimental. So we want to not have the cells come in. So the immune cells can't migrate in there. And so we're using several different methods, but one of the classic methods to do this is basically just mix it in with a material called alginate, feed it through a needle, dropwise into a gelation bath, and voila, you have these capsules. And so this is an islet cluster here shown in green, surrounded by this bubble of polymer. It's actually alginate, and alginate is isolated from seaweed. So it's this material that kind of encapsulates those cells. And they see tremendous results in smaller animal models, smaller preclinical studies, that this is very effective. And a lot of industry got very excited about this potential, like, oh, we could just encapsulate things and then nothing would get rejected. And there's this great paper that kind of talks about all these companies that jumped into this space of encapsulation. And if you pay attention to this big red X, you can see a lot of these companies got out. And the reason for that being is that translation of these ideas from a small animal model to humans has been exceptionally challenging. And that's because mice are just not men. Our immune system, or women, our immune system is much more elegant. And so you can't just simply create some little polymer and the body would be like, I'm cool with that. I don't, that's totally fine. It's this weird polymer, but I'm not going to recognize it. No, it's like, wait, there's something weird going on here. I sense something going on here. And so they will still attack that polymer. So while encapsulation is not a magic camouflage, it can be a very potent tool in an immunosuppressive toolbox. And so it can be effective in keeping the sharks out, but it's not super effective in not stressing out the diver, right? So the islets within this cage tend to be more like me in this diver cage who would be freaking out, as opposed to this diver that seems pretty happy about his situation, right? So if it's not if it's not perfect, how can we leverage this technology better? And that's a big aim in my laboratory, is to try and come up with ways and test and screen ways in which we can use this encapsulating polymer to actually present agents that would instruct the immune cells. So again, going back to this analogy, the shark tank, can we put something on that shark cage that's going to, when the sharks come in and recognize that there's something foreign here, that they will go away? So we have a couple options. They could die, like it could be toxic to the sharks. But that has detrimental effects, because if you're just killing a lot of immune cells, that's bad. We can have it to where they just go away, but then more sharks might come back. The best thing that we can do is convince the shark that this cage is acceptable. And so that's some of the things that we're trying to do is add these agents that would, when immune cells come there, they will recognize this as being foreign, but then they'll get instructions or cues saying, no, this is cool. And so this seems, maybe this is a weird science fiction thing, but this is actually what happens in pregnancy. Think about the fetus is this foreign fetus there. And the placenta, from the moment of egg implantation, actually starts all these immunological pathways that stimulate a protective environment. And so we're actually looking at natural immunoprotective mechanisms that the body has that we could potentially harness. So we grab some of those instructive cues, link it to the polymer itself, and then have that be more protective. So that's an area that we're really exploring and trying to investigate. And the last thing I'll talk about very briefly is one of the challenges that we have in trying to develop new therapies and new approaches for type 1 diabetes is we're really limited on the tools that we can use to evaluate whether something is effective. So really we have two key tools. We have static culture. So we just basically put a bunch of cells together, we mix them together and say, is this working? Is this stopping immune activation or things like that? Or we have animal models. And animal models, while very useful, there's ethical challenges of using using animals for study, and also they're not humans. And so a lot of things that we're learning from models that may be effective are difficult to translate. As I just talked about with encapsulation, it works so well in mice, but it doesn't necessarily work well in humans. And so what we're trying to do is improve culture systems. And we have the capacity to do this, creating human cell sources because of all this advent in cell technology. So you may have heard things like pluripotent stem cells. We can collect skin samples from people and actually create stem-like cells that we can then make a repertoire of a bunch of different types of cells. So we can create beta cells. We can also create immune cells. And so if we can do that from the same person, we can put them together and have them kind of interact. But we don't necessarily want them to do this just in a static, boring, artificial culture. We want to do that in one in which we can create a more environment that has 3D matrix, that has flow, that more mimics body. And so I'll show you a little example of taking islets and immune cells and putting them in this 3D matrix. Can everybody hear me okay? It's saying my internet's unstable. I hope everything's okay. So we can put this in a 3D matrix and look at the cells interacting with each other. So this is actually a mouse islet shown here, this kind of gray blob here. The red are dying cells, so there's just the surface of the islet. And you'll see these little green cells interacting with the islet. And these little green cells, as you can see from the video, are kind of crawling around onto the islet surface, migrating around, sampling and interacting with it. And so you see this very dynamic window. And if you look at these little cluster of dead cells here, you'll see in a minute, this little immune cell will come around and actually eat it. Just chew that right up. So you can see a lot of these immune processes. So this is a really cool window into kind of these immune interactions that we don't necessarily have the ability to do in standard culture or in animal models. And this is just a study of human ones. So these are human immune cells just coming in and just tearing apart these beta cells to try, so this is a way of inside and looking at destructive responses. And this hopefully will give us another tool to think about, well, how do we develop different immunomodulatory materials? Very multi-collaborative project. I do want to highlight, it's all about team-based research. So we have immunologists, cell biologists, other engineers, biosensory experts, and other people that are kind of coming together to allow us to really screen and test effectively human cells within a more dynamic 3D environment. To my most important slide, which is acknowledging my amazing, amazing team that works so hard in the laboratory and really have just been such amazing, resilient people through COVID and all the challenges that they faced with that, and worked so hard in our laboratory, and the diversity and of thought and perspective that they provide is really pushing science forward, as well as our collaborators, which are so important to create multidisciplinary teams that really allow us to bounce ideas off each other and think in a very multidisciplinary perspective to address this extremely challenging problem, as well as finally our funding resources. JDRF has been very supportive of our work, as well as NIH and NIDDK. So with that, I'll be happy to take any questions and chat with you about some of the work that we're doing, or about engineering, tissue engineering, or tissue engineering and diabetes. Thank you.
Thank you, Cherie, for that fascinating presentation. We have an opportunity now to have some questions. If you could put your questions in either the chat or the Q&A section, I'll be happy to moderate the questions for Cherie. And thanks for soldiering on in the midst of your internet connectivity problems. You did great there, Cherie. Thank you.
I hope it wasn't too bad. I'm like on my cell phones.
No, that's amazing. Worked well. Thank you. So while we're waiting for questions, I have a question I wanted to ask. So you're obviously very interested in translational science. What would you say between the time you start an experiment to the time when a patient could sample that therapeutic or that technology, even let's just say in a clinical trial, what is that period of time typically for these kinds of treatments?
That's an exceptionally challenging timeline. So, from the work that we do of developing a new idea to validating it, bless you,
Thank you.
validating it in extensive preclinical models, that usually takes about 8 to 10 years. And then from that time point where you submit a dossier to the FDA, that takes another, to start clinical trials, that usually takes another 5 to 6 years just to start the first human patient. So it tends to be a 15, 20-year process, which can seem very daunting. But, this research takes time, and evaluating safety and efficacy is really utmost importance. But we've made so much progress so far. So I'm not just my lab, everybody has made so much progress. So it's really exciting to see all the dynamic changes and all the things that we have available. The things that I was talking about with the diabetes on a chip, the cells that we're playing around with right now are cells that didn't exist 5 years ago. We didn't have the capacity to make them. So the insight that we're gaining and hopefully being able to develop better therapeutics and a better approach is one that I'm extremely excited about.
We've gotten some compliments for your work in the chat. It's amazing work, impressive to see how this can give hope to diabetics. There have been a couple of questions about artificial pancreas. How does this relate to the artificial pancreas?
That's a great question. So the artificial pancreas is the concept of basically having an insulin pump and a glucose sensor that interact with each other. So doing it through a device-based approach. And there's been tremendous advancements in this, and the insulin pumps and the glucose sensors have just come such a long way. And I think you can consistently, with people with type 1 diabetes, have been very grateful for all these advancements. Of course, there's some economic challenges with those. It's very expensive. So there's some gatekeeping there that we need to address for medical insurance and support. But basically what happens is you have an insulin pump that delivers insulin into the subcutaneous, so under the skin, or potentially into peritoneal space. And then you have a glucose sensor that kind of sits on top, so senses blood sugar under the skin. So this works pretty effectively. So the sensor will sense the blood sugar, and then the insulin pump will release amount of insulin. And there's a lot of things that play around with insulin release, different types of insulin, and ways of that to try and create very stable throughout the day. What happens when someone eats, though, however, is they have to program that in, right? So the insulin pump doesn't know, oh, you're ready to eat. And what are you going to eat? You're going to eat a hamburger versus, 10 cookies or something. They don't know that. So you really have to tailor that and customize the amount of insulin you give yourself to make sure that it's controlled very well. And then the big challenges with these sensors is it's sensing under the skin, not in your blood. So it's not as dynamic. So there's always kind of this lag between when you give yourself insulin through the pump and when it actually kind of gets into the circulation and then gets sensed by the glucose sensor. So there's a lot of engineering process control that goes in there. There's a lot of engineers that really made significant progress on that, but you'll never have, from the artificial pancreas, you'll never be able to have kind of the physiological control that a native pancreas can provide or that cell therapy can provide. And you won't be able to kind of eliminate that mental burden. A lot of people that have these artificial pancreases are kind of overwhelmed by the data. They just get so much information that it's hard to manage. So we'd like to have it to where they kind of receive this cell therapy and they don't think about that they have diabetes for a very, very long time.
Next question is there similar work being done on type 2 diabetes?
Yeah, so there's a lot of similarities between type 1 and type 2 in terms of the end stage. So while the pathology is very different, it's more of insulin, the beta cells become exhausted and insulin absorption issues. There is a lot of similarities to some of the work that we're doing in replacement of cells and other things. When someone with type 2 is at the stage where they're relying on insulin, they could be eligible for cell therapy if we're able to create a more safe, accessible approach. So the lessons that we're learning from type 1 and cell therapy for type 1 could be applied to type 2 for those that are insulin dependent. Not all type 2 diabetics are insulin dependent, but that could be an option.
We have a number of technical questions as well. What techniques do you use to formulate the scaffolds, and what materials are they made from?
Okay. So the technique that we use to make the scaffold is it's a particulate leaching technique, which is just a fancy word. So basically, we use silicone, which silicone has a very bad reputation for with silicone breast implants, but silicone is actually one of the most ubiquitous materials that are used in medical devices. It's used for pacemaker leads. It's used for so many different applications. It's a very pliable, stable material that can be used. And the reason why we selected it is we wanted a material that had a good safety profile for long-term implantation in the context that we're using it in as a kind of three-dimensional support. It also is used a lot for drug delivery. So different implants that you can put, hormonal implants and other things, that uses silicone as a release mechanism. So we wanted something that could allow for local drug delivery. And how we fabricate it is we basically just take the silicone pre-curing, we mix it with salt. So we have salt of a very distinct sizes. We use these sifters like gold, sifting for gold, but we sift for different size salts. And we mix it together to form this paste. We compress it into under high pressure. And then the silicone gels and then or cures. And then you can leach out the salt by just bathing it in water. So you can get rid of the salt. And when the salt goes out, you have this nice porous scaffold. We also have new methods that we're using in actually 3D printing scaffolds. So we're kind of 3D printing these instead of using salt. And that gives us a little bit more control over the porosity, the smoothness of it. So it gives us a little better control of that. So that's a lot of what we're playing around with now. But we've shown that this silicone in this structure has been very compatible with the body. Good question.
Can you say something about the variety of autoantibodies which cause the death of beta cells?
The variety of antibodies.
Autoantibodies.
I'm believing for the Q&A. Okay. There's chats and then there.
Yeah.
Yes. So yes. So there's I don't have a slide on it, so I can't really show it. But basically, when somebody comes in with type 1 diabetes, typically they're at the end stage of over 80% of the beta cells are lost. And they're presenting with hyperglycemia and typically very, very high, dangerous levels of hyperglycemia. But there is a smoldering immune response that happens before then that leads to this destruction. And so through new studies and a lot of what UF is contributing to, as well as other USF and others, is trying to identify genetic risk. So identifying people that may be at genetic risk for type 1 diabetes. Now, some don't have any genetic risk at all and they develop it. So it's not a perfect method. But if they have, say, a close relative with type 1 diabetes or things like that, they may be eligible for a screening. And basically they'll screen the blood to see about the development of these antibodies. And typically what happens is you have these autoantibodies that develop that are kind of flags for an autoimmune condition going on. And so you can have one or two or three or several of them. And it's unknown kind of what, which ones are the primary ones or how these kind of develop. Some suspect that as the beta cells are dying, and they're dying in these very dramatic fashions, they're releasing these things that stimulate more autoimmunity and stimulate these antibodies. So there's still a lot of research going on in that, but there is a very strong correlation between autoantibodies and the development of the disease that helps address some of those questions.
Has there been any research into whether an autograft of beta cells derived from the pluripotent stem cells from skin samples you mentioned reduces the autoimmune response compared to an allograft?
That's a great question, Jordan. Yes, so we have all this amazing technology now where we could take skin cells from a person with type 1 diabetes and in theory, create beta cells from that person using pluripotent stem cell technology. And then we can infuse it back into the patient. From that perspective, you reduce the immune rejection from the fact that it's the patient's own cells. So you get rid of what we call alloimmunity, which is when you transplant between two different people, but you'll still have the autoimmune response. Whether that's more aggressive, it's probably very similar. And so that's a lot of the studies that are going on. And there's people now trying to create stealth cells. So basically, if we can genetically engineer the cell to not express the flags that identify you as self, that may reduce it. But I think in the end, we're still going to have an autoimmune response because the body's still going to recognize that beta cell is a beta cell. And so we're trying to come up with ways to re-instruct the immune system to no longer recognize the beta cell as being foreign is really important to that. But yes, it does eliminate a lot of the allo responses if we can do that.
An endocrinologist in Jacksonville asks, in addition to glucose uptake, insulin suppresses lipolysis and increases lipogenesis. Insulin travels through the portal system. Will the Omentum model allow adequate concentration to the liver? Also, are all of the hormones produced by islet glucagon, GLP-1, etc., produced in your models?
Yeah,
hopefully I pronounced that correctly.
That is great. Thank you, Ken, for your question. Yeah, so one of the reasons why we like the Omentum is it is portal drainage, which basically means that if you think about your digestion, everything kind of shuttles to the liver. So things when you eat, all the nutrients, a majority of the nutrients get shuttled kind of into the liver, where the liver is your metabolic powerhouse. It helps regulate all those things. And so the pancreas is kind of in stream of the liver. And so that's why it can have this intimate control of sensing glucose and secreting the appropriate amount of insulin. And that's likely why cell therapy works when you infuse it into the liver. This islet works pretty well when you put it in the liver because it's in the liver now. But there are other challenges with putting it in the liver. Why we like the Omentum is this portal drainage. So it drains to the liver. And so we believe we can get better control, better sensing of glucose and secretion of insulin to get circulating insulin very effectively than we would if we transplant in a different site. So say if you put it under the skin, it's got to pick up the blood vessels that are in the skin, and then it's got to go through the circulation. And so there's this lag. So we want it to be an intimate communication with the liver. as you're kind of highlighting, Ken, to be able to have that physiological control. And yes, we transplant the whole islet, so not just the beta cells. So it does have the other hormones there. And we believe that that's important so that they communicate with each other in the regulation. We know that it's beyond just dumping out of insulin. These cells kind of really work together to do that.
Any thoughts on the development of glucagon that can be delivered via pumps so that a person can control BG both ways?
Yeah, that's a great question. So there's a lot of people working on, this evolution. So once we had the insulin pumps and then they're still not perfect, can we add glucagon, which is another hormone that plays a really key role in glucose regulation. And so there are researchers that are looking at creating these dual pumps that deliver both insulin and glucagon so that they can work synergistically together to control blood sugar. For our transplants, we're transplanting the cells that are whole cells. So the alpha cells are there, so they'll secrete glucagon as well.
Is there any research into finding which islet cell proteins initiate the immune response and use of a viral vector to disable that part of the islet cell DNA prior to injection, of course?
That's a great, I mean, you guys have got some great ideas. Yeah, that's a big part of what people are trying to study. And this is the limitation, is that when you think about the human disease and the development, we only get discrete time points. And that's a very painful thing to collect. These are from cadaveric donors where people donate their pancreas, and then we can look at what's kind of happening, but it's a very discrete time point, right? So usually these are processed, you get a section on a slice, and then you kind of look at it and say, what's going on here? So it's difficult to know what that initiating factor is. You can't really, study somebody from birth and see what's going on in their pancreas in time, or collect things from their blood and know what's kind of happening. Animal models can provide some insight to that. So there is autoimmune models, there's mouse autoimmune models, but the progression of that disease is very different than the progression in humans. And so there may be some discrepancies there. So while we're learning some of these kind of key initiating events, potentially, there's still a lot to be learned. And that's why we're particularly excited about this diabetes on the chip model, is we could potentially take a healthy beta cell and immune cells that maybe have this genetic component that makes them susceptible to autoimmunity. We can put them together and see what maybe those initiating events are, so that we can then try and mitigate that. So that's a very lofty goal, but that's really trying to develop the platform so that others can really study that.
Why are the separated islets introduced into the bloodstream of the liver and not to the pancreas itself?
That's a good question. So for the reason why they do it in the liver is twofold. One is, as we all know from our Greek mythology, Prometheus, our liver has tremendous regenerative properties. And so you can lose a large portion of your liver, although I don't recommend it, and it will regenerate. And so when you take these islets and you put them in the bloodstream and they lodge into the microvasculature of the liver, what happens is